So if we look here, which is microscope images and schematic representations of antibody epitope recognition, here you could see that the recognition of a pathogen or an epitope is highly specific for the structure of the antibody itself. So here our antibody has a structure that can recognize a small little component of an antigen. And you could see that here as well. You can see here that you can have an antibody recognize sort of a longer cylindrical shape. Here you can see a more oval shape, and here you can actually see an antibody that has an extrusion that will actually go into an epitope to recognize the insides of an antigen, which is really unique and allows for our antibody repertoire to recognize a whole slew of different types of antigens, which is very critical when you're generating an immune response. Now that we've gone through antibodies, let's look at their counterpart, the T-cell receptor. So T-cell receptors are a heterodimer. So right here we could see this heterodimer. And these are very similar to this component of antibodies. So this component of antibodies that looks, this upper portion is called the Fab, or the Fab fragment of the immunoglobulin. So you can see here that the T-cell receptor highly resembles this Fab component of the antibody. And if we dive a little bit into it, it's very similar because if you remember, the antibody recognizes its antigen at this variable region at the top seen here. And this is where the T-cell receptor also recognizes its antigen. Its antigen binding site is here. So T-cell receptors are made up of two different chains. So we have our alpha chain here and our beta chain here. And T-cell receptors are always transmembrane proteins, whereas antibodies are both transmembrane proteins or they're secreted. And if we look at our variable region, the variable region is up top here, and this is what interacts with the specific epitope for which this T-cell is specific. And then we have our constant region here, which includes the transmembrane domain. And this is important as this constant region is critical in leading to the signaling cascades that'll activate the T-cell and differentiate it into an effector cell. In terms of antigen recognition, T-cell receptors and immunoglobulins, or antibodies, recognize their antigens in very different ways. So as you saw, immunoglobulins recognize their antigens kind of when they're floating around and at very specific portions of the full intact protein. Now, T-cell receptors recognize their antigens in a very different and unique way. So epitopes that are recognized by T-cells are often buried within the antigen that we're looking at. So if you can remember here, this is the antigen that we were looking at before. And in the schematic we saw before, we saw antibodies that were targeting this upper portion and this lower portion that were kind of on the outsides of the antigen. However, the epitopes that T-cell recognize are often within antibodies, so very deep inside. So these are often not going to be recognized by antibodies. So what happens is T-cells will actually recognize antigens that are broken down or broken apart. So the antigens must be broken down into small peptide fragments before their cognate T-cells can recognize them. These epitopes that are broken down will then bind to a self molecule called an Mhc molecule, also called a major histocompatibility complex molecule, that'll hold on to the epitope, and then your Mhc molecule, which is on a subset of cells called antigen-presenting cells, so these are cells that present antigen. These cells will present the antigen on their Mhc molecule to a T-cell receptor. And this interaction between an Mhc molecule holding an antigen and its cognate T-cell receptor will activate the T-cell, leading to an effector response. This is a very critical idea, and we're going to delve into what Mhc molecules are in a little bit more detail right now. So let's dive into a little bit more of what Mhc molecules are. So Mhc molecules are proteins that are involved in presenting antigens to T-cell receptors. So these molecules are really important in activating our T-cell receptors. And these are also the most polymorphic genes in the human genome, allowing for these proteins to display as many molecules as they possibly can, such that we have a very diverse T cell response. Now, there are two different classes of MHC molecules. The first class is MHC class 1, and the second is MHC class 2. Now, they're both highly polymorphic cell surface proteins. However, MHC class 1 is expressed on most cells, and there's a very important reason for this that we'll be diving into in a few slides. And MHC class 2 molecules are expressed primarily on highly specialized antigen-presenting cells. MHC class 1 molecules present antigenic peptides that are generated inside the cytosol to specific T cells called CD8 positive T cells. And MHC class 2 molecules present antigenic peptides that are derived from internalized extracellular pathogens that present to CD4 positive T cells. Now let's dive in more into what MHC class 1 molecules are. So if we look at the structure of MHC class 1 molecules, we see that it's consistent of two polypeptide chains, an alpha chain that's encoded by the end of MHC molecule, and an additional protein called beta-2 microglobulin. Now, MHC class 1 molecules, as I mentioned in the previous slide, are fairly ubiquitously expressed. So as you can see here, they're expressed in pretty much every single cell except for non-nucleated red blood cells. Now, this is highly important because MHC class 1 molecules, they will present antigens that have been found from within the cell. So these molecules present peptides that are found from the cytosol of the cell. Now, I'm just going to give you a couple seconds to think about why would you want to display proteins from inside yourself on every single cell? Now, I don't want you to think about it in terms of why we would want to present our own proteins, but are there any sort of reasons why you would want to present something that's maybe not yourself from the outside? What about intracellular pathogens such as viruses and bacteria? Every single cell in the human body or in any mammalian body is capable of being infected by a virus. And viruses are obligate intracellular pathogens. And so when they're replicating inside of our cells, these peptides need to be presented on MHC class 1 molecules so that our T cells that can recognize these viral proteins can be activated. So MHC class 1 molecules are very important in the antiviral immune response and in also presenting any other intracellular pathogens. So if we look at MHC class 1 molecules and the peptides that they present, MHC class 1 molecules depicted here from the top, so if we go back, here we're looking at it from the side. Here at the top is where the peptide binding cleft is. And if we take a downward look at the peptide binding cleft, we see it here. It kind of looks like a hot dog bun, made up of two beta sheets and an alpha helix at the top. So MHC class 1 molecules bind very short peptides that are between 8 and 10 amino acids long. And importantly, I want you to remember that these are proteins that were originally in the cytoplasm that have then been processed and cut up and then presented on the MHC class 1 molecule. So if we look at a sample peptide here, you can see that the binding of this very short peptide is stabilized on each end, such that the peptide is really sitting inside this groove and is completely held by the MHC class 1 molecule to subsequently be presented to CD8 positive T cells. Now, if we look at MHC class 2 molecules, they look similar with an important distinction. So MHC class 2 molecules are actually made up of two different protein molecules seen here, the alpha chain and the beta chain. And MHC class 2 molecules, unlike MHC class 1 molecules, are expressed on a small subset of cells. So they're expressed most highly on B cells, macrophages, dendritic cells, and some epithelial cells of the thymus. Now, these cells, the ones that express MHC class II molecules, can also be called antigen-presenting cells. So these are cells that present antigens on their class II molecules. Other nucleated cells, such as neutrophils, kidney cells, neurons, they do not express MHC class II molecules. The reason for this is because MHC class II molecules will present peptides that were intracellularized or phagocytosed from the extracellular milieu. This allows for immune responses to be generated against extracellular bacteria or any other invading pathogens that are not intracellular. Now, if we dive into, again, looking at that upward look of MHC class II molecules, you can see that it's very similar to our MHC class I molecules, but they have a little bit more opening on either end. This is because the length of peptides that are bound by MHC class II molecules is actually longer and it's not constrained. MHC class I molecules have a very short and specificity of their peptides. They must be between 8 and 10 amino acids long, but MHC class II molecules do not hold this specificity. And they display, again, these proteins that are intracellularized from the extracellular milieu. And you can see that although there are some points of contact which stabilize this longer peptide, in the peptide binding cleft of the MHC class II molecule, they're not bound at the ends, which allows for much more flexibility in what peptide can be expressed. Now, I know I was mentioning CD4 and CD8, positive T cells, but what does this mean in actuality? So T cell receptors have co-receptors, and these co-receptors are called CD4 or CD8 cells, or CD4 or CD8 receptors. So CD4 or CD8, which are, although it's seen here to be on the same cell, they're never expressed on the same cell once these cells leave the thymus. CD4 or CD8 associate with their TCR on the T cell receptor surface, and they bind to invariant sites on the MHC portion of the MHC molecule, allowing for a much more stable interaction between T cell receptor and MHC receptor. I realize I just said a complicated sentence, so let's dive into that. So CD8 cells always interact with MHC class I cells. So why this is important, let's dive back a little bit. So as we've learned before, CD8 cells, CD8 positive cells, are cytotoxic cells. This means they kill the cell that is expressing the protein for which they're specific. Now, MHC class I molecules, as I've mentioned, express intracellular peptides, so proteins that were found in the cytosol. So when MHC class I molecule is expressing a particular protein, so let's say, for example, this cell here was infected with a virus, the MHC class I molecule will take portions of the virus coat or the inside and display it on itself. When a T cell comes in contact with an antigen with a cell that is infected or has an MHC class I molecule, CD8 will actually form interactions with MHC class I, allowing for a tight interaction. Then, this schematic is a little bit different, but we have our T cell here, our antigen-presenting cell here, our T cell receptor here, and our CD8. So again, we have our MHC class I molecule, which is loaded with a viral peptide, our CD8 stabilizing the interaction between the MHC class I molecule and the T cell receptor and the T cell. This stabilization between CD8 and MHC class I allows for tight interactions between the MHC molecule and the T cell receptor molecule. This allows for the cytotoxic T cell to subsequently kill the cell that is infected. And this is really critical in minimizing the cells that would be generating large amounts of viruses or any other intracellular bacteria. So this, what's important to remember is that CD8s T cells are activated by MHC class 1 molecules. So this is a rule called the rule of 8. So you can remember that 1 times 8 is 8. Then, if we look at CD4 cells are activated by MHC class 2 molecules. So CD4 times the MHC class 2 is also 8. This is also the rule of 8. It's an easy way to remember which MHC and CD molecules interact with each other. If we have this concepts between the CD4 MHC class 2 interactions are very similar and parallel to those between CD8 and MHC class 1 interactions. So you can see here that the MHC class 2 molecules can interact with the CD4 molecule. And as before, if we look at a different schematic of what's happening here, we have our T cell and our antigen presenting cells. Our T cells are expressing CD4, and a T cell receptor and our APCs, or antigen presenting cells, are expressing MHC class 2 molecules. Now, if you can remember, the MHC class 2 molecules are expressed by a small subset of antigen presenting cells, which are subsequently presented to CD4 positive T cells, and subsequently these CD4 cells are activated. Now that we have a very broad introduction to adaptive immunity, so the functions of T cells and B cells and the structure of their receptors, I want to very briefly go over a concept that will be delved into in a little bit more detail in future lectures. And this concept is that the adaptive immune system is actually activated by the innate immune system. And this is actually fairly logical, because if you can remember from before, we were discussing that the adaptive immune response is activated predominantly when the innate immune system is unable to contain the invading pathogen. So we want to have signaling between the innate immune system to the adaptive immune system so that they can coordinate their responses. Now, a very critical player in bridging the innate and adaptive immune responses are the dendritic cells. So dendritic cells really form this bridge between innate immunity and adaptive immunity and mediate the transition between the two. Now, this really occurs in secondary lymphoid tissues. So as you've learned previously, secondary lymphoid tissues is where adaptive immune cells are activated. So immature dendritic cells that are within peripheral tissues will migrate through lymphatic vesicles into regional lymph nodes. And within these lymph nodes, the dendritic cells, which are antigen-presenting cells, which are carrying various different antigens on their MHC molecules, will then interact with T cells and other cells in the lymph nodes to subsequently activate these effector lymphocytes. This is a very brief 2-slide overview of this bridge, but it's a really important concept to keep in mind throughout future lectures until we dive into the main details of how this interaction occurs. And with that, we've really encompassed our brief introduction to adaptive immunity, where we talked about lymphocytes, so our T cells and our B cells, where T cells come in two flavors: T helper cells, which mediate the type of adaptive immune response that we have and can also directly kill infected cells. And we also talked about B cells, which are either express B cell receptors on their surface or secrete these molecules as antibodies. We also talked about the clonal selection hypothesis, which states that lymphocytes that are specific for an antigen will be activated and differentiate and proliferate in response to the presence of that antigen, and then will act to eliminate that specific antigen. We then talked about the different structures of antibodies and T cell receptors, that they both have constant regions and variable regions, where the constant regions dictate their function and their variable regions dictate their specificity. We had a very brief introduction to MHC molecules, and one of the main take-home points is that CD8 positive T cells recognize antigens on MHC class 1 molecules, and these MHC class 1 molecules tend to express antigens that are found within the cytosol of cells, and MHC class 1 molecules are fairly ubiquitously expressed on most nucleated cell types in the body. And CD4 T cells recognize antigens on MHC class II molecules. And MHC class II molecules are expressed on what are called antigen-presenting cells. So these are predominantly macrophages, B cells, and DCs. And MHC class II molecules will present longer peptides that are internalized from the extracellular milieu and broken down inside the APC. And then we very briefly touched on how adaptive immunity is activated by the innate immune system, and that these adaptive immune responses are initiated by APCs and secondary lymphoid tissues. And that concludes our brief introduction to the adaptive immune system.